Live streaming is on.
So we start in 1 minute. I'll check again the sound. Yes, it works well now. Good morning, good afternoon, good evening. So we are broadcasting from the TU Delft for this session on xenobiotics, xenogenetics, horizontal gene transfer and antibiotic resistance within the MIWI conference. We did an opening before about what we want to address over the day about water and environmental health protection. And we have the honor to have Michael Gillings from Macquarie University. He's a professor of molecular evolution and had a series of very nice articles and critical opinion articles about antibiotic resistances, xenogenetic elements that are disseminated in the environment, and about the evolution, genetic evolution of antimicrobial resistance in nature. So we will have the keynote of Michael, followed by 5 presentations of 10 minutes contributed from the abstracts, and then a roundtable discussion as usual with on-site online participants. So we are able to make it semi-hybrid for your knowledge, Michael. So there are people on-site, people online who may interact with you. And if you can stay the whole session with us to join the roundtable in the end, this would be fantastic. So we tested your screen before, so you can share your screen and then we can listen to your presentation and the floor will be yours.
Thank you very much for that kind introduction. And my name is Michael Gillings, as David has said. I'm coming to you from the traditional lands of the Wallumattagal clan of the Darug Nation, and I'd just like to pay my respects to their elders past, present, and emerging. So today I'm going to talk to you about antibiotic resistance genes, and I'm going to do so from an evolutionary, environmental, and ecological perspective. So let's share screen here. Okay. So a slightly different title from the one that was advertised, but the principle is the same.
We can see your screen, we can hear you very well
Good.
as well.
That's excellent. So I'm going to talk about how water connects environments, humans and animals, and how antibiotic resistance genes hitchhike on that water to spread to different environments. So the first thing that I want to say is that antibiotic resistance really is a global health crisis. The O'Neill report in 2016 suggested that deaths from antibiotic-resistant infections will reach 10 million a year by 2050, and that will eclipse deaths from cancer. So this is a serious problem, and 2050 is one of those years at the moment. We've got to get to net zero by 2050. We'd like to have antibiotic resistance under control by 2050 as well. So where does this resistance come from? Because antibiotics are molecules that inhibit bacterial growth, and to do so, they usually target particular biochemistries or particular features of bacterial cells. So one way that bacteria can become resistant to the effect of antibiotics is to accumulate a mutation in the gene that encodes the antibiotic target. So for instance, if the antibiotic targets bacterial ribosomes, a mutation in one of the genes for ribosome construction can lead to resistance. But mutations inside bacterial lineages they're a problem, but they're not a really serious problem. And the reason is that that mutation is inside that bacterial cell, and the only cells that are resistant are the cells that arise from that particular cell dividing. So, it's in one particular strain of E. coli, and that's it. A more important problem is where genes are acquired for resistance by the process of what's called lateral gene transfer. And this is where bacteria pick up genes from other bacteria or from the environment. And this is critically important for controlling antibiotic resistance because lateral gene transfer confers immediate resistance. Bacterium steals a gene from somewhere and immediately it's resistant to the antibiotic, and it can spread between different species. And so this is what I'm mainly going to be talking about today, lateral gene transfer of antibiotic resistance genes and how water mediates that process. So when we think about resistance by lateral transfer, we know that we can get genes transferring between all sorts of different bacterial cells in all sorts of different ways. And natural environments contain hundreds of thousands of genes that could potentially confer resistance. For instance, bacteria have things called efflux pumps in their membranes. These pump out compounds from inside the bacterial cell. Some of these efflux pumps can pump out antibiotics so that if you grab a gene for an efflux pump, you can then pump antibiotics out of your cell and you become resistant. But of course, those efflux pumps, their original function was not for dealing with the high concentration of antibiotics that we use in medicine or animal husbandry. They're for something else, some environmental compound that bacteria want to pump out of their cells. It just happens that they can pump out antibiotics as well. So if you can grab a gene for an efflux pump, you become resistant. So lateral gene transfers from the environment occur all the time, and they've occurred over the entire history of bacterial evolution. But when you get transfers into humans and domestic animals and companion animals that are being treated with antibiotics, it fixes those transfers. So one cell in amongst all the cells in your gut will pick up a resistance gene, and it will have an enormous advantage over all of the other cells in your gut, and it will grow, and a large population of those cells will now be resistant because they have this laterally transferred gene. And so our antibiotic therapy has actually been very efficient at fixing these rare lateral transfer events into bacterial pathogens and commensals that are of importance to humans and agriculture. Resistance to antibiotics appears really quickly, and it seems to be inevitable. So we know that lots of resistance genes have been acquired by lateral gene transfer from the environment. And we also know that that process is pretty fast. Usually within about a decade of introducing an antibiotic, so here's the antibiotic timeline of when various antibiotics were introduced, and when resistance was first observed is underneath here, and you can see that resistance often appears within a decade of the introduction of an antibiotic class or type, and often it's much quicker than that. So resistance appears to be inevitable, and so what we need to do is to think about the processes that lead to resistance, and in particular lateral gene transfer and how it works. So an example that I've been working on for a long time is called the clinical class 1 integron. Now, integrons are genetic elements that scavenge genes from the general environment. They captured and express genes like they try genes out and get rid of them if they don't confer an advantage and keep them if they do. So integrons are really exquisitely pre-adapted to capture antibiotic resistance genes from environmental organisms and from environmental DNA. The clinical class 1 integron has an additional advantage because it jumped onto a mobile DNA element, a transposon, which then jumped onto a plasmid and then was able to transfer between all sorts of different bacterial species. We've worked for a long time on reconstructing the origin of this first clinical class 1 integron, and we now have evidence that it arose as a single copy, like a single genetic event in the early 1900s. And from that initial event, which occurred at a single location, in a single cell, in a single human, somewhere in maybe the 1920s, this class 1 integron on the plasmid has now acquired over 130 different resistance genes. It's spread to a whole series of mobile broad host range plasmids that move between different bacterial lineages. As a consequence, it's spread to over 70 species of bacterial pathogens. And in fact, it's the major cause of antibiotic resistance, gram-negative pathogens. It has now spread to every continent, and it has become a universal feature of the microbiota of humans and agricultural animals. So you and me and all the other people that are listening to this talk right now, we all have class 1 integrons in our gut microbiota, and those class 1 integrons carry resistance genes. And as we get older, so this is the diversity of resistance genes in gut microbiota, as we move from being a child to a school student to a high school student to an adult, the number and diversity of resistance genes in our gut microbiota increases. Now, as a consequence of that, we're constantly releasing these resistance genes into our homes, into our general environment, and into sewage streams, human waste streams, either directly from humans down sewage treatment lines or from agricultural animals or from companion animals. So here's just an example. We ran a project testing vacuum cleaner dust. 95% of the samples we've looked at are positive for this DNA element, the class 1 integron integrase. And in fact, that gel that you can see there with all those strong positive bands, that is actually from the lounge room that I am sitting in right now. So we shed all of these resistance genes and mobile elements into the general environment, largely through feces. So humans, domestic animals, and companion animals all carry large abundances of these mobile DNA elements and resistance genes. that were not found in humans, domestic animals, or companion animals 100 years ago. But now they are, and they're at high abundance, and we shed these back into the environment, and there they can interact with environmental microorganisms and with wild animals. And much of this transmission is waterborne. As a consequence, resistance genes have become a significant environmental pollutant because water goes down pipes and eventually it ends up in rivers, lakes, and estuaries and the oceans. And as it does so, it carries resistance genes and mobile genetic elements with it. So here's a study where we looked at estuaries along 4,000 kilometers of China's coastline, and every single estuary that we looked at was highly polluted with antibiotic resistance genes at 1 million to 100 million copies per gram of sediment. 100 million copies per gram. A gram of sediment is about the size of a sugar cube, 1 centimetre by 1 centimetre by 1 centimetre. In fact, it's a bit smaller than that. Now, of course, estuaries are where fish and crabs and shellfish grow, filter the water. So these animals are accumulating these resistance genes and mobile elements, and of course we eat those. In 2017, I did a back-of-the-envelope calculation of the numbers of resistance integrons that are shed every day from humans and some domestic animals. So we know how many copies of the class 1 integron there are per gram of feces. We know how many grams of feces each of these animals produces per day. We know the size of the population. So you just multiply all those out, and the total copies released per day run somewhere between 10 to the 18 and 10 to the 23 copies, depending on what animal you're looking at. In this case, pigs being probably the worst. Now, remember that this particular element, this class 1 integron, clinical class 1 integron, was a single copy sometime in the 1930s. So this is possibly the most successful DNA element in the history of the world. Now, that results in significant environmental contamination, of course. So here is a dot plot of the prevalence of this gene in environmental samples, and it's taken from 45 different qPCR studies. So in soil, probably Antarctic soil represents the historical abundance of this particular element. Farm manure, of course, is elevated. Wastewater is elevated. So is sludge and effluent, and rivers and lakes and freshwater sediment. You can see that resistance genes survive water treatment. So this class 1 integron, clinical class 1 integron, actually survives the entire treatment, from wastewater to sludge to effluent to rivers and lakes and into the sediment. That's because wastewater treatment is designed to remove bacterial cells, not DNA molecules and selective agents, which are small. Here's a small study just looking at individual resistance genes. So there are 10 adults in this study, and this is not a high-powered sewage treatment plant. It's a reed bed filtering system, so it's a little bit different from standard wastewater treatment plants, but the principle is the same. So 10 adults. Between them, they had 128 resistance genes, and you can track the abundance of those in the influent to this treatment plant, in the effluent, and then downstream. And you can see that some 53 of the resistance genes that were present in the 10 adults actually survived the entire process. Now, one of the reasons for this is that DNA can survive as naked DNA, it can survive inside cells, it can move from one cell to another, and waste streams often have selective agents in them that promote the survival of cells that contain these particular resistance elements. So the potential consequences of all of this. Firstly, there's a vastly increased abundance of resistance genes in the environment. and that increases the likelihood of lateral transfer into environmental bacteria. It increases the likelihood of acquisition of resistance genes by wild animals. So here's just a panel of wild animals that we've found resistance genes in: prawns, wallabies, flying foxes, sea lions. These all have resistance genes in them that are typical of the type of resistance genes you find in clinical contexts in humans. So those integrons and resistance genes and plasmids are still active. So they can acquire novel resistance genes with selective advantages from the environmental resistome. In other words, there's multiple possibilities for further evolution. And what this does is generate ever more complex, what I call xenogenetic DNAs. These are DNAs that would have never been seen on the planet but for human attempts to control bacterial growth. They're often complicated multi-compartment molecules with lots of different resistance genes in them, not only to antibiotics, but to disinfectants and heavy metals and other resistances. And they're very complex DNAs that have been assembled from all sorts of different original bacterial locations. Okay, so all environments are connected by water. The terrestrial environment is connected to other bits of the terrestrial environment by water. It's connected to estuaries, lakes, it's connected to the ocean. And you can think about this as cycles where hospitals, livestock, human populations, pharmaceutical industries are all dumping waste into either agricultural land or wastewater treatment plants. Those go through soil or into surface waters and aquifers. The surface water and aquifers are used, come back as drinking water or as on the surface of plants or through recreational exposure back into humans and our agricultural animals. So that's a bit of a problem for us. We need to start thinking about all of the different compartments. For a long time, antibiotic resistance has been thought of as being a hospital problem. It's actually an environmental problem and an ecological problem, because human waste, sewer overflows, effluent, sludge, septic systems go into the environment and contaminates soils, sediments, and water. Wildlife waste and livestock manure does that as well. And then we get water-related exposure, ingestion of contaminated drinking water, recreational exposure, consumption of food irrigated, washed with contaminated water. So we've got a cycle here of all sorts of resistance genes moving around, generating more complexity, picking up more resistance as they go. Water management is the key to controlling antibiotic resistance. We do need to understand the magnitude of the fluxes of resistance genes and, importantly, selective agents that are going into water systems. And we're going to need to develop new surveillance techniques for these to detect resistance genes, mobile genetic elements, the presence of antibiotics, heavy metals, disinfectants, and things like that in water systems that we manage. So DNA, in this case resistance genes, joins other pollutants as a cause for concern. And I have a fairly broad view of pollution. I think it's the dissemination of materials that could have harmful effects. So, conventional pollutants, crude oil, carbon dioxide, heavy metals, nitrates. Xenobiotics are things that humans generate, pesticides, plastics, synthetics. I would also class invasive species and these xenogenetic DNAs as pollutants as well. So invasive species disseminate from where they should be, are transported somewhere, and then cause problems. Xenogenetic elements, complex mobile DNAs, resistance genes, and here I'd also include synthetic DNAs and the DNA that comes out of genetically modified organisms. The difference between conventional pollutants and these invasive species or xenogenetic DNAs is that these are pollutants that can replicate. And they replicate, when they do replicate, they confer advantages to organisms that are exposed to other pollutants, such as antibiotics, disinfectants, heavy metals. So where do we go to from here? Well, we kind of need to rethink the way that we treat water, because we need to remove selective agents from waste streams. And we also need novel treatments of wastewater to reduce pollution with resistance genes. Now, it's not just resistance genes, because the next phase is virulence genes and pathogenicity genes and viruses and phage and all of the other things that go through. And we certainly need a much better understanding of the dynamics of these genes. Where do they come from? How long do they last? How many are there? How are they affected by selective agents? Where are those selective agents coming from? How can we minimize the effect of those selective agents in maintaining high abundance of resistance genes and other genes in our wastewater streams? So human attempts to control bacterial growth are good because it means we don't die. But like many things that humans do, they've had these adverse consequences that probably weren't predictable. So selection for resistance genes and selection for increased movement of those genes via lateral gene transfer. Assembly of these complex mosaic DNA elements that carry multiple resistance determinants. Rapid increases in the distribution and abundance of these DNA elements, largely from pollution via wastewater streams. Widespread pollution of these genes now and their host cells in environmental compartments, and contamination of ecosystems with selective agents. So we definitely need to consider both the ecological and environmental aspects of antibiotic resistance, and one of the key threads that joins all of these environmental compartments together is. So thanks for your attention. I am happy to answer questions either now or in the panel, or let me see. Oh, yes, there I am. Or if you want to, just email me after the conference is over. Thank you very much.
Thank you very much, Michael, for this inspiring talk on antibiotic resistances and xenogenetic elements. And so we have time for questions, since you were quick with your keynote. So it's very nice, so we can interact with online on-site delegates. We are chairing this session together with Frances Corbera-Rubio and Sergio Tomas Martinez here. So are there some questions to Michael, online, on site? Yes, Sergio?
Hello, thank you for the presentation. I have one question. When you talked about the appearance of resistant genes in wild animals, so I guess that the wild animals are not like subjected to antibiotics. So how do these, like the bacteria there, maintain these genes? So what is the advantage that it confers to them?
So what we've discovered is proximity to humans increases the frequency and abundance of antibiotic resistance genes in wild animals. So often, if wild animals are being treated, by vets, or if they're in a zoo, or if they're close to human habitation, they have more resistance genes. Okay, so one of the things that I mentioned during the talk is that, of course, all of these elements are still active. And so, for instance, I showed you a picture of a prawn, and we found in the guts of those eastern king prawns various species of Acinetobacter, not the ones that are emerging pathogens, but potentially they could be. And the integron in that case has picked up some genes that confer resistance to oxidative bursts by the animal immune system. It repairs damage to proteins. So the bacteria has picked up a gene that actually stops it from being damaged by the animal immune system. And often there's not a great cost to housing these elements, at least in the short term.
Thank you, Michael. Can I ask you to maybe unmute your video so we can see yourself as well?
Oh, I am unmuted.
Or
I'm not
to
quite sure.
The camera.
the camera. Yeah.
Yeah. Hold on. I can see me. I'm up there in the top corner. So and the camera is unmuted, so I don't know where it is. I can see you guys sitting around a table.
Yeah. We see your acronym, so maybe our colleague Dryfta can solve the problem.
Yeah, I don't know.
Okay.
I just unmuted and put it back on again, but yeah. Sorry.
Okay. But no problem, we continue like this, yes.
Yeah.
And so how would you define this term xenogenetic DNA? So if we can recall, try to give a clear definition to the person, what is xenogenetic DNA?
So the Greek prefix xeno means foreign. And so when you talk about xenobiotic compounds, you're talking about chemicals that humans have synthesized that have never been seen on the planet before. And so these are brand new compounds that are not naturally occurring. I coined this term xenogenetic as a parallel or an analogy with the xenobiotic, because these complex mosaic bits of DNA that have resistance genes, metal genes, transposons, metal resistance, disinfectant resistance, lots of different resistance genes all stuck together. All of those genes have come from separate places, from different bacterial donors. And so they've been assembled into these long, complicated mosaic DNA elements. And none of those elements would have ever appeared on the planet except for the fact that humans have imposed all of these selection pressures on bacteria with disinfectants, with heavy metal pollution, and with antibiotics. So that's the kind of long version of what xenogenetic is. It's a piece of DNA that is brand new on the planet and has been generated by human activity.
Thank you. There are some questions in the chat, Francesc.
Yes, there's a couple of questions. The first one from Mr. Shangzi Wang. Thank you for your amazing keynote lecture. What do you think about the use of meta-omics to assess lateral gene transfer? Do you think extra well lab experiments should be done to verify this transfer? And last, what other mobile genetic elements are critical other than integrants?
Okay, so probably the most critical elements are transposons, because there's a lot more of them than there are integrons, and integrons need transposons to move around between plasmids and between hosts. So controlling transposition is a really important thing. The use of metagenomics is great. I mean, I have nothing bad to say about metagenomics at all, but it's not very good at detecting rare events, and it's not very good at detecting rare genes. So, for instance, if we do metagenomics for integron gene cassettes, we very rarely find resistance genes, even though we know they're there. And this is in pristine environments. So I think metagenomics is a fantastic tool. It has to be used for the right question, though. Certainly, comparisons of mobile DNA elements between different taxa, between different phyla of bacteria, and even between different domains of life, it's easy enough to show that things have transferred because the sequence is identical in 3 or 4 different organisms that are actually not closely related. And so that means there's been a recent lateral transfer. But of course, by the time you detect that, it's too late because it's already transferred. What we really need to be thinking about is controlling this whole process, figuring out how lateral transfer works, what stimulates it, and what environments it's most likely to occur in, and then try and manage that.
Okay, thank you very much. There's another question from Niti. Awesome presentation. What would you suggest in terms of treatment process to address AMR? Which are your recommendations on pretreatment or tertiary treatment?
Yeah, that's a really good question. So I am not an engineer and I'm not a water specialist. So I'm hoping that the talk that I gave will stimulate people in the audience to think about, okay, what is the best way to remove these agents and genes from water flow? I don't know whether some kind of membrane filtration, reverse osmosis, I don't really know the technical details. Whatever it is going to be a bit expensive, but if 10 million people a year are dying, that's a worthwhile thing to think about. There's also other potential things like ozonation or biological treatments. I actually don't know because this is not really my field. I just recognize that there's a problem here, and I could help in detecting the success with which you remove DNA, but I wouldn't know where to start about detecting removal of antibiotic residues, for instance.
May I ask a follow-up here?
Yeah, sure.
Yes, I'm Mark Vloosdrecht, Delft University. In principle, it's not a problem to remove all DNA from, say, wastewater. The technology is there. But what is needed is how much should we remove in the end, also in a kind of environmental and economic terms. That needs a kind of approach which is similar to the quantitative microbiome risk assessment, which is there for pathogens.
Yes.
Are you aware of any developments within the antibiotic resistant gene spreading of a kind of quantitative risk assessment methods?
Okay, so yes, there are. There's a group that has been meeting every second year called Environmental Dimensions of Antibiotic Resistance. And they've been thinking about these minimum concentrations. For instance, they've thought about the no effect concentration of antibiotics, not the minimum inhibitory concentration or the minimum, this is the minimum no effect concentration. So you have to get it down to that or roughly there. They've also thought about applying the same kind of strategies of, we need this many genes per mil and below that we're safe, like this many E. coli per mil or crassphage or whatever. They have been thinking about that. It is difficult to do, partly because, you have to wait for people to die of antibiotic resistant infections and yeah. But yes, you're absolutely right. That is what's needed, is some quantification of the risk versus the cost that you need to expend in order to get down to that level. It's always going to be a balance between those two.
And in this context, on quality criteria, so in wastewater treatment plants, we generally measure that in the per liter of effluent of wastewater treatment plant, we have around 10 to the 6, 10 to the 7 copy numbers per liter. Is it high? Is it low? So you give values in a table here to say that per gram of feces, you have 10 to the 6 to 10 to the 11 copies of this class 1 integran. How can we get a sense of these values in terms of concentrations, in terms of number of gene copies? When we speak about xenobiotics, like pharmaceuticals, we know that we should not go above 1 microgram per liter, maybe in European norms for some pharmaceuticals. How can we put a number on?
Okay, so this again is a really difficult problem because of the way that quantitative PCR is done, and often in quantitative PCR, it's expressed as copies per copy of 16S ribosomal RNA. So if you have 1 copy of 16S ribosomal RNA per mil, you've got no bacteria in the water at all, but maybe that 1 bacterium has 10 copies of resistance genes, and so it would look huge because, you know, the ratio is 10 to 1. We really need actual quantitative estimates of these in terms of parts per million of genes of interest. And again, this is somewhere where I'm not really qualified to talk. I can do the assays, but the if you like, the accreditation of measurements is beyond my expertise. It's a really good question though, and it is something that I hope people will be stimulated to think about after listening to the talk that I just gave.
Thank you so much, Michael. Is there a final follow-up on site on Colin, maybe? Yeah. And we keep the discussion for the roundtable after. Yeah.
One last question from Colin Norton, which will give a keynote session this afternoon. There were 2 questions. Yeah. What percentage of antibiotic resistance is mutation, and which percentage is lateral gene transfer?
Oh, that's a really good question. So lateral gene transfer is by far the most important, simply because it can move between different cells and different species, whereas mutation is restricted to the cell lineage in which it arose. So we have, I would say that, you know, the things that we really should be concerned about are lateral gene transfer. And the numbers that I've been giving on numbers of resistance genes, number of integrons, those kinds of things, all of those numbers are genes that are subject to lateral transfer. So I think lateral transfer far outweighs mutation as a concern. And don't forget that once you get a mutated gene, it can then find its way onto a mobile element and then be laterally transferred. Yeah.
Thank you so much, Michael. This is really interesting to discuss with you and to have your knowledge, and if we can benefit from it for the roundtable after the.
You don't need to come to office. You just share your ID, right?
Somebody
Okay,
should
so-
mute the microphone online. Sorry.
Thank you
Right.
very much for the questions, and I will talk to you at the roundtable. Yes.
Thank you so much.
Thank you.
And I give the words to Francesc for the next part of the talks. Yeah, we are used to this nice congratulation signals. Yeah, it's 3 days of amazing. Exactly.
Thanks again, Michael. There are many questions for you, so I approved them. You can find them in the webpage in Dryfta. And let's start with the oral presentations from the abstracts. The first one is from Ryo Honda from Kanazawa University in Japan. And we will launch the video now.
Turn it on again.
My name is Ryo Honda from Kanazawa University. Thank you for coming to my presentation. My presentation is about profiling and international comparison of antibiotic resistance in activated sludge by metagenomic data analysis. Antimicrobial resistance is known to have locality, and the MR in a country is determined by local factor, a history of antibiotic use, and international factor, which is traveling of people, food trade, and so on. So to know the determinative factor of AMR in a country, it is important to understand the global scale.
There are some problems for some people. I think that they cannot see the video. We are trying to solve it.
I can't believe how many problems I've had with all these technical things. You think that like after the meeting would end, they would sit down and say, okay, look, we need to sort this out. This has to work tomorrow.
No, this is crazy. It's the third day. And I mean, they should have had technical takes to see that
Yeah.
it works.
And that's the whole reason that we were supposed to send our recordings, right? So they could get everything.
And it's also not the beginning of the pandemic, so they had a lot of time.
Yes, this is right. Yeah, exactly.
I have a feeling they're not fully in control of, like if it was just a regular Zoom meeting, but I think it's because they
Mute the microphone, please.
at the last minute.
Somebody has a microphone open, please stop it, and we try to mute it as well.
Yes, I'm gonna go ahead.
Let's launch the pre-recording again. Sorry for that.
Hello, everybody. My name is Ryo Honda from Kanazawa University. Thank you for coming to my presentation. My presentation is about profiling and international comparison of antibiotic resistance in activated sludge by metagenomic data analysis. Antimicrobial resistance is known to have locality, and the MR in a country is determined by local factor, a history of antibiotic use, and international factor, which is traveling of people, food trade, and so on. So to know the determinative factor of AMR in a country, it is important to understand the global scale AMR distribution. Antibiotic resistance in wastewater was investigated by metagenomic approach recently. However, the major source of AMR spreads into water environment is not only untreated wastewater, but also the wastewater treated effluent, which is associated with the resistance of activated sludge. So the objective of this study is to understand international diversity of resistance in activated sludge and to reveal antibiotic resistance genes, which is unique and characterizing the resistance of the country. We compare the resistance of 4 countries, which is Japan, US, China, and Denmark. The metagenomic sequence data in Japan was collected by wet approach. Wastewater and sludge samples were collected at 5 wastewater treatment plants, and metagenomic sequence data was obtained by shotgun metagenomic analysis. The sequence data from US, China, and Denmark was downloaded from DDBJ and the ENOA database. Finally, we could get 10 sludge sequence data from each of 4 countries. And for wastewater, we collected the 10 sequence data from Japan and 6 data from US. The sequence data was blasted with CART database to get the ARG profile of the sample. The ARG profile includes the list of ARGs and read count of each ARG fragment. The read count was normalized by gene length by using RPK. Then our ARG composition was obtained by calculating proportion of each ARG by using the total RPK of all ARGs of the sample. And abundance of the ARG was calculated by using the 6NS genes RPK. Results of ARG abundance. Abundance of ARG in the slash was the lower than slash. So in any countries, the ARG abundance slash was lower than wastewater. And we compare the antibiotic resistance of these samples. From principal component analysis, the samples were grouped by sludge or wastewater. So resistance in sludge among country, resistance in sludge among country was less diverse than in wastewater. Next, we compare the resistance of only sludge samples among countries. We could find the 3 clusters: Denmark, Japan, and the cluster of US and China. The Denmark cluster was explained by positive PC1, and Japan cluster was explained by negative PC1. And negative PC2 explains the cluster of US-China. So we can explore the characterizing ARG of each cluster from PCA loadings. Here's Japan is characterized negative PC1. So ARG with highly negative loadings are here, and here is the ARG with highly positive loadings characterizing the Denmark. So resistance of both countries were commonly characterized by resistance to macrolide, quinolones, and the cephalosporin. However, the genes encoding these resistance were different. In Japan, the mostly multi-drug efflux were characterizing their resistance. In Denmark, ESBL genes of OXA and the IMP or efflux pump were characterizing the resistance. And the US and China clusters were characterized by ARGs to lincosamide, glycopeptide, and ESBL-TEM genes. And we couldn't distinguish US and China from PC analysis, but from the PLS analysis, partial least square regression analysis, we could extract the distinctive ARG of US was peptide resistance genes, ARMA, and the lincosamide resistance genes. Conclusions. So resistance is primarily defined by sludge or wastewater rather than by country. And the resistance of sludge was less diverse among countries than wastewater. Antibiotic resistance of the 4 countries were grouped by 3 clusters, and we could find unique ARGs abundant in each group. Thank you very much for your attention. Bye-bye.
Thank you very much, Rio. Are you online?
Yes, I am.
Hi. Good evening. Are there some questions to Rio? Maybe one question from my side. So what drives these differences in this cartography of resistant genes between Japan, Denmark, USA, and China? So on your last slide, on the conclusion, you highlighted the main differences, but what is the cause behind these differences? Is it the use of the antibiotics themselves, or does it originate from somewhere else?
Thank you for questions. So yeah, we are still investigating that, but I think one of the big impact factors is the use of antibiotic, and this is because the sludge resistance is affected by the wastewater, and the wastewater is much affected by the clinical use of antibiotics, and we couldn't include that in the presentation, but we compared with the clinical use of antibiotics between Japan and the US. And the sludge and the wastewater resistance in US, it's in the lincosamide resistance genes are uniquely abundant. And we didn't find that in the Japan resistance. And when we compare the clinical use, that the lincosamide was used like 4% of the total prescriptions. And in Japan, it was almost negligible. And the example is the cephalosporins. It was highly abundant in the Japan in clinical use data, and cephalosporin resistance is more abundant in resistant genes, more abundant in wastewater in Japan than US. So probably the antibiotic clinical use antibodies is one of the big factor, but some drug resistance is independent of that. So I think it's not a single impact factor, but for some resistance, some drug resistance, that is a big impact factor.
Thank you very much. Is there a question in the chat?
Yes, there's a question from Arnaud Deschênes, who will also present today. Thanks for sharing your results. Have you explored genes associated with mobile genetic elements? If so, are there any differences across countries?
Yeah, thank you very much. Yes, it's still ongoing, so we haven't shared the data, but we are doing that, and we want to find some association with the ARG and difference among the countries. So I hope we can report that maybe soon. Thank you very much.
Okay, I suggest that we move forward to the next speaker, and you will stay online for the roundtable. So we'll come back to you. Thank you so much, Rio.
Thank you very much.
So the next speaker is David Calderon Franco from Delft University of Technology, who will present on site. And the title of the presentation is Transfer Dynamics of Resistance Determinants, Microbiome, Resistome, and Mobilome Profiling of a Full-Scale Granular Sludge Wastewater Treatment Plant. Let's go.
Just to check, can we see online? On YouTube as well. Yes.
Hello, can you hear me? Great. Then good morning, everybody, or good evening, good night, wherever you are in the world. My name is David Calderon Franco, and I'm going to present my project on transfer dynamics of resistant determinants, microbiome and resistome and mobilome profiling of a full-scale granular sludge wastewater treatment plant here in the Netherlands. First of all, and I guess you have seen this graph quite often, and you're going to see it quite often today, I want to talk about the One Health approach and antibiotic resistance. And as Michael Gillings has said before, antibiotic resistance, it's a multidimensional problem that's happening right now, and that involves a variety of different actors, such as humans, like us, agriculture, farming, or the natural environment per se. And it's based on the assumption that bacteria and genes can move freely around them and spread between them. But we are in a water conference, so that's why we want to focus on wastewater treatment plants. And why wastewater treatment plants? And One Health, because basically water is what is joining all of these actors, and humans, we have generated anthropogenic barriers, such as wastewater treatment plants. So it's interesting to understand what's the effect of such barriers on the transfer and spread of antibiotic-resistant genes in the environment. So, as I said, I work on granular sludge for this project. So here we have a very nice figure of a granule. And the question is, why why granular sludge as an environment? And why aerated granular sludge as a plant? So what I wanted to focus on my project was that until now, most of the analysis that has been done and checked during the wastewater and antibiotic resistance has been focused on the intracellular DNA, which mainly is mainly people are checking how bacteria are being removed from wastewater treatment plants. However, little is known about what is the effect or what happens with the free-floating extracellular DNA that is being released due to cell decay or other phenomena during the process. And this DNA we are interested in because in the overall project of my PhD, we want to study how natural transformation happens and how this DNA can be transferred to other microorganisms. Moreover, there's little known about what happens with antibiotic-resistant genes in different metric biomasses and biofilms. So that's why aerated granular sludge is a very nice environment for it. We don't know which mobile genetic elements are they using for transferring and mobilizing. And one of the main questions that everybody asks normally in all the conferences and meetings, like who are them, who are the potential hosts, and how can they uptake it? That's why we thought that studying a differential analysis of the aerated granular sludge would be the perfect ecosystem to differentially study the fate of extracellular DNA and antibiotic-resistant genes in the environment. For doing that, let's see if it appears here, we took an aerated granular sludge tank here in the Netherlands, and we differentially isolated intracellular and extracellular DNA from both the influent inside of the tank, which included also different kind of biomass sizes, like big granules, small granules, and floccular sludge, and also in the effluent. And what we checked from it was basically we quantified the overall effect of the process in terms of resistant load, and also we analyzed the microbiome resistome and movilome of such samples. Next. So, as we don't have that much time, I'm going to just highlight a little bit, some results that we came for and which bottlenecks we faced depending on the samples that we got. So first of all, I wanted to talk about the microbiome analysis. We are working with aerated granular sludge, which is a complex sample that not everybody, there's not that much of history, let's say, in terms of sequences uploaded and published. So when we use standard databases such as Kraken, we found that the annotation rate was quite low. So for improving a little bit and the annotation, we use the MIDAS field guide. We retrieved the 16S RNA sequences from our metagenome data, and we tried to complement it also with some literature research, and we added all these extra genomes that we found that were present in a MIDAS field to the Kraken 2 database. And this is only like, I just want to show this, and I don't want you to go into all of these numbers and names. It's a lot. So we can see in the green box, like this was before we adapted the database. Those are the microorganisms that we could find. And then when we added the MIDAS database into our database, we could see that these bacteria in the red box were not present before and appear more than 3% when the database was adapted. So this kind of analysis helps to understand better our systems and to give messages such as follows. Like we could say, for example, like the extracellular DNA originated, in our case, mainly from the microbial granules during the aeration process. So this could be seen because we had the adapted database. And if we compare, with the other, so we have here extracellular DNA on the first two, and then 1 and 2 basically is the replicates that we had. So it's the same sample from different days. we could see that the extracellular DNA was similar to the granules, unlike the flux, which was more less annotation rate and also more diverse sample. Then we could see, we can say that using this kind of approach that differentially isolating different DNA fractions and different kind of biomasses, that we can study or understand phenomena such as cell decay, immigration that we heard yesterday in different presentations, microbial dynamics during the processes. Something more that we can say about this kind of analysis, we study the microbiome, but we are also interested on the resistome. What we can see is that antibiotic-resistant genes, and there's a wide variety of them during the whole process. But what I wanted to highlight here was that in the effluent of the wastewater treatment plant, there were 478 different antibiotic-resistant genes, so the variety was quite high, sorry. 90% being on the extracellular DNA fraction. And this I will come later with when I talk about the qPCR results, which is like how overlooked and how hidden has always been this fraction and how important we think it is. But before we go there, and moving on a little bit more, what's the question that everybody's asking? Who are these potential antibiotic-resistant gene carriers? And we know there are better techniques, let's say, for analyzing this. We have the high-C sequencing or other kind of techniques for doing that. But we basically use this approach that I want to explain. So we took the contigs from our whole metagenome containing antibiotic-resistant genes, and we selected which one contain antibiotic-resistant genes, and we re-annotate them with this adapted database that I was talking before to get kind of a list of potential microorganisms that could carry antibiotic-resistant genes. And this list basically is this one. So we have Pseudomonas and Rhodophyrax mainly as the potential carriers of antibiotic-resistant genes in our samples. And this is something, especially Rhodophyrax, that I have already seen, I think actually in the one of the last papers of the Repares workshop. I wanted to say that the majority of them are gram-negative, and Pseudomonas being one of the most critical ones, already explained or highlighted by the World Health Organization. So important to highlight this. Then, just to move on, another one of the things that we were interested in, that Michael Gillings has highlighted before, is to try to understand these antibiotic-resistant genes. Where are they embedded in? What are they using to move around and to transfer between microorganism and microorganism? So we tried to do following a similar strategy as I have said before, a colocalization study analysis between mobile genetic elements and antibiotic-resistant genes. So we took all the contigs containing antibiotic-resistant genes and mobile genetic elements in the same contig. And also from those, we only selected those contigs that contain more than one antibiotic-resistant gene and a mobile genetic element, which is this MGE term here. And what we could see is things like this. Okay, we got a list of 420 or 450 mobile genetic elements containing antibiotic-resistant genes across the whole process. But I just wanted to highlight that this is how they look like more or less when we try to retrieve them. So we see that at the end of the reaction of the process, we have some of these constructions, but what I wanted to highlight were mainly the ones in the extracellular DNA in the effluent, which are the ones that potentially can retransfer and reintegrate in other microorganisms. So doing this kind of colocalization studies are interesting, I think, because maybe using long-read sequencing would be even better to completely narrow it down. But we can understand, if we can reconstruct these plasmids with the genes, if such plasmids are narrow or broad range. And also we can understand if such plasmids are natural occurring or are synthetic. So we could, for example, try to sequence, which is the multi-cloning site, which is something that humans have synthetically generated. And then for finishing, just to have an idea of what was the overall process of the plan, I did some quantitative PCR analysis. And what we can see here is that we just check different genes, and maybe it could be other approaches, but this is what we chose. There's a significant reduction of antibiotic-resistant genes over the process in general. That in all phases, and that's something we already saw before, makes sense. The intracellular DNA contains much more antibiotic-resistant genes than the extracellular DNA. That the genes that have the highest concentration are sulfonamides, which also makes sense because it's the more historically antibiotic used. But then Another thing we checked was what was in general, the variation between the influence and the effluent of such genes. And what we can see, is that intercellular DNA, which is basically bacteria, is released during the process in general. So that's what we have seen in most of the already published papers. So wastewater treatment plants are effective somehow at reducing the load of antibiotic-resistant bacteria. But what we have seen on the other side is that on the extracellular DNA fraction, this is less consistent. So we see some genes that are increasing, some genes that are decreasing, and this we should put a focus on this maybe. So yeah, what I said before, that the extracellular DNA fraction is the only one containing genes that increase over the process, even though we would need validation and more sampling campaigns that we have seen before that would help proving this point. So in general, I would say that, plants, in this case, aggregated granular sludge, are effective at reducing bacteria, but due to the process and cell decay and other mechanisms, there's like a release of DNA in the environment. So to conclude and not to steal more time, I want to say that this differential analysis of the intercellular, extracellular, different biomasses, it's very interesting to study the processes such as cell decay, immigration from sewage, and microbial dynamics. That the extracellular DNA fraction in the aerated granular sludge effluent originated mainly from granular sludge. That the majority of the antibiotic-resistant gene carriers were detected were gram-negative. And that the colocalization studies are very interesting and important to understand where the plasmids come from and where they can go. And regarding the quantification, as I said, the extracellular DNA is an underestimated and overlooked fraction that I think I want to highlight that we should take a look on that. But there's a significant reduction in terms of bacteria over the process carrying antibiotic-resistant genes. So with this, I finish, and I want to acknowledge my master student, Roel Sarelsen, Stella Christo, who helped me doing the analysis, to Mario Pron, who helped me design the experiment and the sampling campaign, and all my supervisors, Mark, Thomas, and David, for the guidance over this PhD. Thank you very much.
Thank you so much, David, for your presentation with clear messages and to the point. It's not easy to express all this data that you generated from the plant. Quick question, would you think it would be different in an activated sludge system than in a granular sludge system?
Regarding what?
What you observed in this granular sludge or aerated granular sludge plant, do you think that you would see the same in a conventional floccular activated sludge system or not?
So you mean like a release of DNA in the environment? Sorry.
Yes, so what I understood from our study is that the aeration process or the process itself leads to lysis of bacteria, potentially, and release of extracellular DNA that is not so abundant in the influent, it's highly abundant in the effluent. It's the primary part that has the genes. So do you think in a conventional plant, this release of extracellular DNA would be the same or different than sequencing batch reactor design with granules?
So I didn't do a sampling campaign like the one we did for this aerated granular sludge tank, but the method that we developed for isolating the extracellular DNA was done checking from 3 time points activated sludge plants. So actually it was developed with activated sludge plants. And we could see actually that the extracellular DNA fraction in the effluent of activated sludge plants was significantly lower than the influent, but also containing high amounts of antibiotic-resistant genes. So I would expect similar behavior.
Okay, and what are the questions in the chat, Francesc?
There is time for one question from Mr. Changzi Wang. Thanks for your amazing presentation. For 11% of the antibiotic-resistant genes not in the extracellular DNA, do they belong to a specific drug family? And also, how did you identify transposons on contigs?
Hi, thank you for the question. They don't belong to a specific family, only to one family. So what we could see was that both the extracellular and the intercellular DNA fraction, when we reannotated such contigs, so they were all belonging to those 10 gram-negative families that I was saying, like in highest abundant. They were also belonging to other families, but in the highest abundance were mainly the 10 that I have highlighted before.
Thank you very much, David.
Thank you.
Let's keep the other questions for the discussion panel. And now we move to the fourth speaker of the session. It's Fuzhong Xiong from Peking University. And the topic is use of quorum quenching bacteria to control biofouling and spreading of antibiotic resistant genes in membrane bioreactors treating antibiotic containing wastewater. We'll launch the video.
Hello, it's my great honor to have the opportunity to give the oral presentation in this conference. I'm Fuzhong Xiong, a PhD candidate from Peking University. My topic today is about the use of quorum quenching bacteria to control biofouling and spreading of ARGs in membrane bioreactors. The spreading of ARGs has been a constant health concern for years. Especially in China, the antibiotic consumption was quite huge, and the ARG burden per capita was also extremely heavy. Many researchers consider that the biological reactors in the wastewater treatment plants might be the hotspots and black boxes for ARG dissemination, because in these areas there were high-density microbial cells and continuous subinhibitory antibiotic pressure. These conditions are favored for horizontal gene transfer of bacteria in these different forms. Among different wastewater treatment technologies, MBR has advantages in removing ARGs and ARB, for it can effectively retain bacterial cells and DNA fractions by filtration. But these ARGs and ARB were transferred to excess sludge, so the risk still existed. Another constant challenge for MBR technology is its fouling problem. Recently, many studies confirmed that the application of quorum quenching bacteria, which can inhibit quorum sensing pathways by degrading QS signals like AHLs, can be used to control the biofouling problem. And there are also studies indicating that QS may affect the horizontal gene transfer in bacteria. So we wanted to know the use of quorum quenching bacteria can be used to control the spreading of ARGs. Therefore, we set up 4 identical lab-scale reactors to explore this question. The whole running period was divided into 4 stages. The first stage is a startup stage lasting for 10 days. And stage 1 was from day 1 to day 7. And in stage 2, we added a mixture of 3 kinds of antibiotics. They are SMX, CFX, and ATM. For reactor 1 to reactor 3, the adding concentration of each antibiotic was 100 microgram per liter. And for reactor 4, the adding concentration was 1,000 microgram per liter. In stage 3, we inoculated QQ bacteria into reactor 2, reactor 3, and reactor 4 with 2 kinds of doses. And the QQ bacteria were entrapped in the semi-permeable membrane bags like this to avoid the direct change of the microbial community in these reactors. And the QQ bacteria used in this study was QX01, and it is isolated from the activated sludge of a real MBR system. During the running of these reactors, we collected sludge samples and fouling samples regularly. And we also designed a procedure to extract different DNA fractions from these samples. The first is cell-free extracellular DNA, which existed in SMP contents. And the second is cell-attached DNA, which existed in APS contents. And the third is intracellular DNA. Then we use high-throughput qPCR to analyze the ARGs and MGEs in these different forms of DNA. And the microbial community was analyzed using amplicon sequencing method. Here are the main findings of our study. These 2 figures show the transmembrane pressure profiles in different stages. In stage 1, the fouling cycle was 7 days in the 4 reactors. And in stage 2, the fouling cycle gradually reduced to 2 days in reactor 1 to reactor 3. And the fouling cycle gradually reduced to 1 day in reactor 4. So we can say the antibiotic pressure significantly accelerated the biofouling processes. In stage 3, the application of Cucubacteria elevated the severe MBR fouling problem caused by antibiotic addition. And we can see this kind of change in fouling cycle in these 3 reactors. However, this effect didn't last for long during the operation of these reactors. It might be due to the decrease of the QQ activity of the inoculated Cucubacteria. Moreover, the extracellular protein and polysaccharide and PN to PS ratio in sludge samples and fouling samples were also analyzed. We can see in stage 2, the EPS content increased sharply in all these 4 reactors. And in stage 3, the EPS content showed a decreasing tendency in reactor 2, reactor 3. We also found that the PN content in sludge EPS had a strong correlation with fouling rate. So the protein content may contribute more to the fouling process. And we detected 4 kinds of AHLs in the reactors. And according to the Pearson correlations, we found that carbon 4 HSL and carbon 8 HSL may play more important roles in regulating SMP or EPS production. So how did the microbial community structures change in the 4 reactors during the whole running time? This figure presented the time decay patterns of the microbial communities in the 4 reactors. And we can see the succession rates of the 4 reactors in stage 2 were higher than those in stage 3. And in stage 2, the succession rate of reactor 4 was higher than the other 3 reactors. And in stage 3, the succession rate of reactor 2 and reactor 3 were higher than that of reactor 1 and reactor 4. So we can find that the inoculation of QQ bacteria can also influence the succession rate of the microbial communities. And we also use a random forest analysis to identify the contributions of microbial communities. Here means top 15 genera to the following related microbial behaviors, like EPS content and SMP content and AHL concentrations. The results show that the microbial communities explained very well for these factors. So we can conclude that the microbial communities also contributed to the MBR fouling processes. More importantly, we analyzed the abundance of ARGs and MGs in different DNA fractions. From this figure, we can see that the addition of antibiotics sharply increased the total relative abundance of ARGs and MGs in the cell-attached DNA. And after the inoculation of QQ bacteria in these 3 reactors, the relative abundance of ARGs in AEDNA of these 3 reactors were much lower than that in the reactor 1. And the QQ bacteria also inhibit the increase of ARGs in intracellular DNA in the reactor 2 and the reactor 4 compared with reactor 1. But their effects on the cell-free DNA were not obvious. Moreover, the absolute abundance of ARGs in cell-attached DNA were comparable with that of ARGs in intracellular DNA. So we cannot ignore this proportion of the total ARGs. And we also wanted to know if the variation of ARGs in these reactors correlate with the HGT processes. So we first analyzed the total relative abundance of MGEs with the relative abundance of ARGs in different samples. And the significant positive correlations between MGs and ARGs were found in the sludge samples and also in the intracellular DNA of fouling samples. And furthermore, we conduct procrustis analysis between ARGs and microbial compositions. The M-squared value of the reactor 2 and the reactor 3, which were inoculated with QQ bacteria, well, lower than that of the reactor 1. So compared with reactor 1, stronger correlations between resistant and bacterial genome in reactor 2 and reactor 3 indicate lower HGT frequencies. So the QQ manipulation might play roles in the horizontal gene transfer of ARGs. So here is a brief summary of the conclusions of the study. And we demonstrated that the QQ manipulation has the potential to influence the variation of ARGs in the MBR reactors. And many thanks for your attention. I'm more than glad to have your kind advice.
Thank you, Fusheng. Are you online? If you have been able to connect online, or if you or your
Yes.
supervisor is online.
Yes, I'm online.
Thank you so much. I have a question. Can you explain us a bit what are these quorum quenching bacteria? What does it mean, this quorum quenching? What are the type of bacteria that have this metabolic process? And what is the mechanism behind the statistics that you give in your talk here?
Okay, thank you for your question. Quorum quenching exists in bacteria because the first mechanism for bacteria communication is the mechanism of quorum sensing, that bacterial cells use signal molecules to communicate with each other and may regulate many microbial behaviors of the bacterial cells. But quorum quenching is a phenomenon that maybe the bacteria can degrade these signal molecules like AHLs. So it can block the quorum sensing pathways for these bacterial cells. So it can affect many microbial behaviors of the other bacterial cells. So there are many studies recently to use this kind of quorum quenching bacteria to control the MBR falling. And we also isolated a QQ strain in the MBR system. So we want to know why we use this kind of bacteria to control the biofouling, and if it has a effect on the horizontal gene transfer of the ARGs, because there are also studies that indicating the QS pathways may also affect the horizontal gene transfer in bacteria. So we want to combine these two phenomena together.
Thank you. And is there a question in the chat, Frances?
Yes, there is one question from Mr. Ziming He. Very interesting talk. Is there a specific reason behind using quorum quenching bacteria instead of quorum quenching compounds? And if so, which are the benefits?
Are you still here, Fusheng? Else we come back at the round table with the question again. Yeah. I think you are muted, Fusheng. Maybe the line is not stable. I don't know. Well,
it says connection lost.
Okay. Well, let's move to the next talk and we come back to this for the round table.
The fifth speaker of today is Zigang Yu from the University of Queensland, Australia. And the topic is non-nutritive non-nutritive sweeteners can promote the dissemination of antibiotic resistance through conjugative gene transfer. We will also launch a video.
My name is Zhigang Yu from Australian Centre for Water and Environmental Biotechnology, formerly AWMC, the University of Queensland. Today, I will give a talk about spread of antibiotic-resistant genes and how non-nutritive sweeteners affect spread process. It is well known that antibiotic resistance has been a global health issue. The emergence of antibiotic resistance can be due to the misuse or massive use of antibiotics that are commonly used for the curative therapies. However, these antibiotics can also drive the development of resistant bacteria and even shift gut microbiota composition. This alteration in gut microbiota composition can be also associated with individual diet and finally affect the human health. As the diet additives, artificial sweeteners have been safely permitted as table sugar substitutes with low calories but high intensive sweetness. So they are also called non-nutritive sweeteners. They are widely popular among individuals who suffer from glucose intolerance and type 2 diabetes. Currently, these sweeteners are widely applied in all aspects of our lives, such as food and beverages, and so we see soaring consumption globally. This consumption really raises a concern about whether they are really safe to our health. A growing body of studies have reported that these sweeteners are associated with some adverse effects, such as weight gain, cardiovascular disease, cancer, and as well, these sweeteners are also able to induce glucose intolerance and alteration of gut microbiota, which is very similar to the antibiotic. So this inspired me to think whether there are some similarities between artificial sweeteners and antibiotics. Previous research have reported that these sweeteners can show antimicrobial effects. As well, they also contain antibiotic-like groups. For example, for the sulfonamide antibiotics, they contain sulfonate functional group. This group can be also observed among the sweeteners such as saccharin and acesulfame potassium. So this raises a question: could artificial sweeteners promote spread of antibiotic resistance in bacteria? So to address this question, I established the 4 conjugation models, including intergeneral, and reverse conjugation models. Especially for the intrageneral conjugation models, I also employ a clinical relevant plasmid, the PMS, which was isolated from a patient who suffered from urinary tract infection. Here, I employ 4 commonly used artificial sweeteners: saccharin, sucralose, aspartame, acesulfame potassium, to do the experiment. Initially, the donor and recipient were mixed together and were exposed to the artificial sweeteners for 8 hours at room temperature. Afterwards, the sample were spread onto 2 types of select plates. The first one was used for the enumeration of the transconjugate number, while the second one was used for the total recipient number. Then we calculate the conjugation frequency based on the number of transconjugate divided by the recipient number. As well, we calculate the fold change of frequency by normalization of the frequency of treated group to control group. Then we did a series of analyses, such as reactive oxygen species production, cell envelope permeability, as well as the whole genome gene sequencing analysis, in order to figure out the underlying mechanisms. In both intra and intergeneral conjugation models, we found that artificial sweeteners can significantly promote the conjugative transfer with a dose-dependent manner. For example, for the intrageneral conjugation model treated by the ASP, at 0.03 milligram per liter, up to 2-fold increase was observed. When the concentration increased to the 300 milligram per liter, up to 4-fold increase was observed. Such increase was also observed for the clinical relevant plasmid transfer between E. coli species. Once successful, received plasmid, the transconjugants are able to deliver such plasmid to the next candidates, which broadens the spread of ARGs. Seeing is believing. We also visualize the conjugation process at a single cell level. Here, we couple the microfluidic system with the microscope, and then we use the E. coli that carry GFP plasmid as the donor. Actually, the GFP gene is repressed by the lactose operant in the host, At the same time, the E. coli, the donor also carried chromosomal tag and cherry gene, so the donor shows the red fluorescence. When successful transfer to the recipient, this GFP can be expressed and enable the transconjugate to be green fluorescent cells. Based on this, we detect the conjugation events and did time course analysis. For the multi-channel images from the control group, we can detect the green fluorescence cells at 10 minutes. By contrast, this exposure to the artificial sweeteners speed up this process, as we can detect the green fluorescent cells within 5 minutes. As well, we also statistically detect the transconjugate number at different time points, and then summarize the results in this table. NM means the transconjugate number, R means the transfer rate. For the control group, we found that around 12 transconjugate number were detected. By contrast, under exposure to the artificial sweeteners, this number also significantly increased. Correspondingly, the transfer rate also increased under such condition. So these results further support that artificial sweeteners can promote the conjugation process. Considering that antibiotics normally can induce the RS overgeneration to pose stress on bacteria, here we also suspect that artificial sweeteners can also induce such similar effects. So we detected the RS production from both donor and recipient and found that the RS production was significantly increased. To further confirm the RS role during the conjugation model process, we did the scavenging test. For both intra and intergeneral conjugation models, we found that after adding the scavenger, the frequency was significantly declined. For example, for the intrageneral conjugation treated by the 300 milligram per liter of SAUK, before adding the scavenger, the frequency was around 7 times 10 to the power minus 4. After adding the scavenger, the frequency was declined to around 4 times 10 to the power minus 4. This means that RS play a critical role during the conjugation process. Such RS production can be also supported by the upregulated expression of RS relevant genes, such as SODA and SODB. As well, we also found the upregulated expression of SOS or stress response relevant genes. This means that our artificial sweeteners can play antibiotic-like roles during the process. For the scavenger test, we can still find that even after adding the scavenger, the frequency was still higher than the control. This means that other mechanisms may also play the roles. Considering that during the conjugation process, plasmid also need to come across the same envelope between donor and recipient. So do artificial sweetener also affect the envelope between them? The answer is yes. We found that the artificial sweeteners can significantly promote the envelope permeability for the donor and the recipients. And such results was also supported by the upregulated relevant genes. As well, conjugation process can be also regulated by some relevant genes responsible for the plasmid replication, transfer, and even the pinning channel for the plasmid transfer. Under exposure to the artificial sweeteners, these relevant genes were significantly upregulated. So based on all these results, I propose conjugation mechanisms. The artificial sweeteners promote conjugation process by increasing RS production and detoxification, activating SOS response, and increasing cell envelope permeability between donor and the recipient, and finally enabling the recipient become resistant bacteria. That's my presentation. Thank you. Thanks for your listening. Take this time, I want to appreciate the conference committee and organizers who gave me this chance to deliver my talk. As well, I also show my appreciation for my advisory team, Jianhua Guo and Ian Henderson, who gave me a lot of support during my study. Last but not least, I also appreciate the financial support from the UQ's Graduate School Scholarship.
Thank you, Shigang. Are you online? Are you or one of your supervisors online? Maybe Jen Hao?
Yeah, I'm online.
Yeah, hi. I have a question about the chemical nature of the these non-nutritive sweeteners, and if there is a link between what you identified on ROS systems with the chemical nature of these compounds. Because if I remember, some of these sweeteners are members of the families of thiazolinones or isothiazolinones that are one of the most important contact allergens for humans. So do you think that there can be a link between what you observe at bacterial level to the allergenic nature of these compounds?
Okay, yeah, that's really good. Thanks for your question. Yeah, so actually, so before answer your question, I just want to figure out the inspiration that why we want to focus on the artificial sweeteners. So the first thing is that the artificial sweeteners are widely applied in our food and beverages, and so this causes another possibility that the people or even other humans can expose to such artificial sweeteners. So what kind of adverse effects could these artificial sweeteners could brought to the humans? Traditionally, these artificial sweeteners are permitted as safe additives. So that's why this is one thing. Another thing is that based on the chemical structures, the artificial sweeteners show some antibiotic-like structures. As I mentioned, that the artificial sweeteners contain some antibiotic-like groups. So based on this, we suspect that the artificial sweeteners can promote the gene transfer. Yeah. Of course, in this study, we didn't conclude that the chemical structure or the compounds can be associated with the horizontal transfer of genes. So I cannot conclude the link between them.
Thank you. Are there some questions in the chat? Question in the room?
Time to move to the last speaker of the session.
Thank you, Shigang.
Okay.
Thank you, of course. So again, it's time to move to the last speaker of the session, which is Tessa Crosby from Rice University in the States. And the title of her work is Harnessing Conjugation Systems for Engineering Microbes and Promoting Bioremediation through the Spread of BPHC Dioxygenase Activity.
Hello, everybody, and welcome to my research talk in which I will be discussing how we have harnessed conjugation systems for engineering microbes and promoted bioremediation through the spread of BPHC dioxygenase activity. Here we have a concept map that outlines our overall experimental procedure in which we inoculate either a single engineered donor or a single synthetic community into a realized soil microcosm. We then follow up by assessing horizontal gene transfer efficiency as well as BPHC functionality stability across time and generation. Finally, we wish to isolate transconjugate species from soil microcosms, extract whole genome DNA, send the DNA for 16S rRNA gene sequencing, and then determine what the host range might be for each of the 5 plasmid backbones that we utilize throughout the study. Polycyclic aromatic hydrocarbon spills have the potential to cause immense environmental damage due to their recalcitrant properties. Certain PAHs, including benzanthracene and benzoate-pyrene, are known human carcinogens. While bioaugmentation of genetically engineered microbes has been attempted, these microbes generally fail to survive and replicate for sufficient periods of time upon inoculation. A potential solution for enriching rather than diminishing engineered microbial fractions in situ involves seeding bioaugmentation with organisms native to the target site. However, few studies have implemented this strategy in dynamic microbial environments. Employing horizontal gene transfer for beneficial purposes, such as for the delivery of functional biodegradative genes, has the potential to advance microbiome engineering and the bioremediation of legacy pollutants. In this study, we focus specifically on evaluating the choice of plasmid backbone on the delivery of a dioxygenase gene, BPHC, to multiple different recipient bacteria via conjugation. We also measure the impact of gene delivery on metabolic performance and the stability of multiple plasmid constructs in a recipient bacteria over several generations. This table shows the different plasmids that were constructed for the study, all of which were engineered with the BPHC gene. These include those ranging from approximately 4,000 to over 65,000 base pairs, have low to high copy number, and some code for their own conjugated machinery while others do not. The POC19 plasmid backbone was further modified to include an RP4 origin of transfer region in order to allow it to experience conjugation events. To deliver functional genes to a microbial community, we focus on using conjugation, a form of HTT that involves the transfer of plasmid DNA from donor to recipient cell via a sex pilus. This sex pilus is encoded by the host's chromosomal DNA, in which case it is non-transferable, or by genes on the plasmid DNA itself. If plasmid DNA encodes for the conjugated machinery, recipient cells become potential donors as they have the genes for the machinery to transfer the plasmid further in a community. This is why HTT has the potential to be a powerful tool for amplifying genetic payloads in complex microbial communities. Here I am showing RSF1010 with the addition of the BPHC gene and its respective promoter and terminator sequences. The BPHC gene codes for biphenyl 2,3-diol 1,2-dioxygenase, an iron heme enzyme capable of ring cleavage activity. The model substrate used in this study is 2,3-dihydroxybiphenyl, a polycyclic aromatic hydrocarbon. Here, DAP-dependent MFD pure E. coli donors electroporated with engineered BPHC plasmids were combined with various recipient species, either on solid agar with a nitrocellulose membrane filter, on solid agar without a filter, and within liquid media for 48 hours. Next, the conjugation mixtures were washed with PBS buffer to remove residual DAP and plated onto an antibiotic selected plate without DAP. This ensures that only transconjugate species appear on the plate. Conjugation efficiencies were then calculated by taking the number of recipient bacteria that received plasmid DNA and dividing it by the number of recipient bacteria inoculated within the initial conjugation mixture. Preliminary results suggest that generally, MFD pure E. coli donor mixed with Pseudomonas butida had higher conjugation rates than when mixed with Bacillus subtilis, except for the case in which PKJK5 BPHC was tested. The asterisks indicate a statistically significant difference between the recipient bacteria's reported conjugation frequencies for each plasmid. Generally, conjugation frequencies were highest when solid agar media without a nitrocellulose membrane filter was used, and these are the trials that we chose to display for the study. Overall, it is clear that the PKJK5 plasmid backbone delivered the BPHC genetic payload at the highest conjugation rate when Bacillus subtilis was used as the recipient species. RSF1010 BPHC was delivered at comparable rates between the two recipient species, and the TP114 plasmid backbone was conjugated at a much higher rate to Pseudomonas butida than to Bacillus subtilis. Now we move on to the second part of the experiment, in which DH10B E. coli transconjugates were tested against their non-engineered native species in terms of substrate removal. Here we measured the removal of the BPHC substrate 2,3-dihydroxybiphenyl by reading the absorbance every 10 minutes for 4 hours in triplicate. These substrate removal rates were calculated by taking absorbance measurements, converting them to concentrations through the use of Beer's law, followed by the normalization of data by OD 600 nanometer readings. Then, each successive removal rate was compared to the removal rate from the day before, and the difference between the removal rates were mapped across the entire serial growth experiment. Here, we subjected DH10B E. coli recipients to media conditions where substrate is either added or absent in media throughout the serial growth experiment. Preliminary data subjecting RSF1010 BPHC and TP114 BPHC DH10B E. coli recipients to a 10-day serial growth experiment demonstrated that media conditions in which substrate was added supported bioremediation longevity across generation and time for both plasmid constructs tested. Furthermore, DH10B plus TP114 organisms far outperformed DH10B plus RSF1010 recipients in terms of specific biodegradation rates throughout the 10-day experiment, despite the significantly larger plasmid size. On average, recipient organisms engineered with TP114 BPHC grown in media alone removed the substrate at a rate 2.9-fold faster than non-engineered counterparts. We also observed an acclimation period that was 4 days for TP114 BPHC and 10 days for RSF1010 BPHC. After the acclimation period, the TP114 BPHC engineered E. coli grown in media with substrate removed 2,3-dihydroxybiphenyl on average 4.31-fold faster than non-engineered counterparts. In conclusion, we suspect that media conditions in which substrate was present supported an offset of fitness burden for plasmid carriage by providing a selective pressure for plasmid maintenance. In conclusion, we found that conjugation efficiencies varied significantly when disparate engineered plasmid backbones were used. We also discovered that bacterial recipient species played an additional role in dictating conjugation rates, with Pseudomonas butida performing better for most trials than Bacillus subtilis and accepting a BPHC genetic load. Furthermore, we demonstrated that plasmid stability is best supported when serial growth media includes the 2,3-dihydroxybiphenyl substrate and after a sufficient acclimation period has been achieved. Overall, we advanced an understanding of how the presence of a unique carbon source in different conjugation systems improve plasmid stability and gene delivery. Future research will focus on assessing conjugation rates and host range of these plasmid constructs to microbial communities as opposed to single recipients. Eventually, we would like to extend this work to longitudinal experiments in which engineered microbiomes are maintained in realistic conditions representative of contaminated sites. Community assembly dynamics conduct the manner in which bacteria interact with one another as well as their surrounding. Thus, an experiment in which substrate is suspended on a microfluidics device or two-dimensional nanomaterial would be necessary to inform computational simulations for capturing realistic bacterial communication networks. Finally, I'd like to acknowledge the Stadler Research Group that is supported by the Rice Civil and Environmental Engineering Program, as well as the National Academies of Science, Engineering, and Medicine. And with that, I am more than happy to take your questions.
Thank you, Tessa. Are you online? Probably it's early morning in USA, or in Houston, or if Lauren or Tessa are online. Maybe not. I don't think so. So I think we should go to the panel discussion now. We continue with this. So if all the speakers could show their camera, and David, you can come here at the table at the microphone. And so we, to summarize what we have seen in this session, we had a very nice keynote lecture from Michael Gillings about the rise and dissemination of antibiotic resistance with the prevalence of these class 1 integrons and the importance to not only look at the genes that confer the resistance, but to look at the transposons as well. Rio Honda expressed an international profiling between Japan, Denmark, USA, and China about antibiotic resistant genes and specific characteristics in the wastewater of these countries, linking potentially to the antibiotics that are released in this wastewater. Then David Calderon Franco spoke about the free-floating extracellular DNA and this importance for gene as gene carrier, as mobile genetic element on antibiotic-resistant genes, and it should not be overlooked anymore. We should not only look at the intracellular DNA in our experiments or measurements, but also the extracellular DNA. And he has a method that can help to differentiate this extracellular DNA from the rest of DNAs that are present in a wastewater and in a sludge. Fusheng Xiong expressed the use of quorum-quenching bacteria to fight biofouling and potentially antibiotic resistance at the same time. And Shikang Yu spoke about the effect of nutritive sweeteners. So they are not only antibiotics that are linking to resistance. Michael also expressed heavy metals and other many compounds that can lead to resistance. And these non-nutritive sweeteners, apparently they have also chemical structure that are similar to antibiotics. And therefore, there may be something there to look at as well. And Tessa highlighted how conjugation systems could be used in a beneficial way to treat complex compounds, notably in soil and in wastewater. And then this brings maybe the question, to which extent can we apply genetically modified organisms in environmental settings, in soils, in water systems? It depends certainly on the local regulations of different countries. So some countries can produce genetically modified vegetables. So how can we apply genetically modified organism in the environment? Maybe, Michael, if I ask you, what do you think about this application of genetically modified organisms to solve environmental services? Do you have any view on this or knowledge about how feasible it is in a non-country?
Okay, so at the moment, I'm a member of the ARC Centre of Excellence in Synthetic Biology. And what we're aiming to do there is to use synthetic biology tools to modify the genome of yeast and then use those yeast strains to value add to agricultural waste and convert it into useful products. but we are not thinking of environmental release of any of these organisms. What we're actually thinking about doing is enclosed bioreactors where you don't get any escape of the engineered organisms. I think, you know, you mentioned that some countries can release GMOs and others can't. I think if genetically modified microorganisms are released in one country, global movement of people, animals, goods, soils, plants will very quickly spread those modified microorganisms all around the planet. Do I think that that's a hazard? Well, yes and no. I'm still on the fence about it. Every single technology has both promise and peril. And our job as scientists is to maximize the promise and minimize the peril. I think that's all I need to say for the time being.
Thank you. And David is also a systems biologist by profession. So what do you think about this application? So you used to work at DTU, I think, in the Synthetic Biology Center there. And how do you think that this synthetic gene modification can be applied to for environmental services or not?
Yeah, I'm a bit in line with Michael. Like, I think the benefits, pros and cons need to be addressed. Not only that, also in case, you know, we were approving such, so it's very cool what the last presentation they have shown. It's quite interesting that we can do that with engineered microorganisms. But in case, let's say, I'm not talking only now about risks. Like, how are we going to, you know, move it on in terms of regulation and controls? Like, how are we controlling this to spread in between different environmental settings? Difficult.
And this thing also to the type of problematic that we have in the Netherlands and in Europe, where there is a complete reassessment of regulations for genetically modified materials and organisms. Antibiotic resistances are part of it as well. And I remember we had a master student who made a small project about how to quantify the risk of a genetically modified organism, a genetically modified material, or an antibiotic-resistant determinant that propagate in the environment. And when we discuss with the National Institute of Health of the Environment and Public Health in the Netherlands, it's not so easy to address the risk of all these different types of materials. So addressing the risk related to a genetically modified material or the risk of an antibiotic-resistant gene is not the same. So synthetic genes are designed, are synthesized, whereas antibiotic-resistant genes, transposons, integrons are still natural features of organisms, but that are, exacerbated by our human activities. So, Michael, about risk assessment, what are the procedures for risk assessment for antibiotic resistance versus environmental bodies or human bodies?
So I'd just like to return to Tessa's talk for a minute. So what Tessa was doing was taking BPHC, dioxygenase, and cracking the aromatic ring using that enzyme. And her approach was to take a known gene and put it into a known transfer agent and then engineer that and then force it into other hosts. There is another way of doing this. Any environment that is contaminated with dioxins or biphenyls or aromatic compounds is already going to have microorganisms that have enzymes that degrade that compound, that recalcitrant compound. The problem is that often the different enzymes are stuck in different cells or different lineages. So we've seen in the talks that we've listened to several methods for controlling the way that DNA moves between different compartments. We've seen the importance of extracellular DNA. We've seen the importance of quorum sensing. We've seen the importance of compounds that induce the ROS and SOS responses. We know how these things work. So if we can control natural lateral gene transfer processes, I see no problem in going to a polluted environment and just giving natural processes a bit of a push. And then, as you say, natural bacterial evolutionary processes will speed up and you will get the right organism to degrade that recalcitrant product fairly quickly. Now, like I said before, peril and promise, there could be downsides to that process as well. So getting back to the risks of antibiotic resistance genes in environmental compartments. So I'm sometimes asked this in seminars. People say, well, look, if everyone has antibiotic resistance genes in their guts, antibiotic resistance genes as a pollutant are not really a problem. And in some ways, that's true. But what is a problem is the complicated mobile DNA elements that arise as a consequence of antibiotic use. So, for instance, we know that there are integron gene cassettes in environmental compartments that can confer phage resistance. CRISPR is seen in gene cassettes that integrons can capture. So also are restriction modification systems that are anti-phage things. So let's say we throw antibiotics out and we now start to control bacterial infections with phage therapy. Already, there are genes out there we know that can confer resistance to bacteriophage. And it's inevitable that bacteria will acquire those genes. What's important is slowing down the rate and the locations at which that acquisition occurs. At the moment, we have so many diverse mobile elements, transposons, insertion elements, conjugative elements, integrons floating around in human-dominated ecosystems that it vastly increases the probability that those rare events will happen. So the parallel here, in some ways, is with COVID-19. So RNA viruses have fast mutation rates. When mutations occur, it leads to increases in transmissibility or escaping vaccines or, all sorts of things that benefit the virus. If we've only got 1,000 virus particles on the planet, the chances of any of those mutations occurring in the right spot at the right time are zero. But if we've got a standing population of infected people, each of whom have 100 million virus particles in them at any one time, the chances of a very rare event occurring and fixing go up hugely. And so while vaccination for COVID may well stop us dying from it, our end game should be to damp down the number of viral particles as much as we can, because that slows the rate of realized mutations as a whole on the planet. The same is true of resistance genes and mobile elements. The more of them we have, the more likely it is that an extremely rare event will occur at the place where the key selection is already in place. So, you know, do I think that a tetracycline resistance gene is potentially dangerous to everyone on the planet? No, I don't. Do I think that tetracycline resistance genes should be removed from water treatment? I don't really think that. What I think is that we need to remove the mobile elements that the resistance genes are on. Would be my long-winded, very long-winded answer to that question.
Thank you so much, Michael. And this makes some interesting links with sessions that we had yesterday on theoretical microbial ecology, where colleagues use stochastic models, notably to address dynamics in communities. If one of the theoretical ecologists is online, like Ezekiel, Tom, or Joe, who presented yesterday, on the others, what do you think that, how could your approaches of mathematical modeling be used to address these transfer events with stochasticity or deterministic approaches? Is Ezekiel or Joe or Tom Curtis online?
Maybe not.
David, can you repeat the question? I was connecting the
On
headphones.
what Michael explained us about we said the transfer of this gene, and we need to remove the mobile gene elements to reduce the rate of the transfer and the localization of the transfer. Is there a way that the theoretical ecological models could be used to make predictions?
Yeah. Okay. I mean, in theory, yes, because you could assess the communities of taxa when you evaluate communities, right, in terms of stochastic or deterministic assembly, and then you could also address communities of genes to see that. But then I guess it gets complicated, right, with regards to these particular genes, because you have elements like horizontal gene transfer and stuff like that. But I think it could be possible. I mean, we just need to put some thinking about it. And, you know, I mean, engineers tend to think that everything is possible, just a matter of paying for it, right? So if we really if we really
You
think
are
of
far
it,
more
maybe
optimistic.
if we really think of it, maybe we can.
In the audience, no. And Joe, what do you want to follow up?
Yeah, so I feel a little out on a limb because ARGs are not my area of expertise, but I'm quickly thinking there's something I should be paying much more attention to. This is really interesting. From my perspective, and I'm in agent-based model land right now, it's theoretically possible to model ARGs and these genetic elements. I know some colleagues of mine are working on implementing SBML, which is a metabolic markup language, into the agent-based model I'm working with. If you can model that phenomena, then yeah, as far as what I've been looking at with stochastic versus deterministic, ARGs are just another fitness factor, and we can then run simulations and talk about, well, where are they prevalent, where are they not prevalent, and how deterministic is this versus just how much of it is a bad luck that you have a ARG enriching environment.
And can we say that if we are able to model in a deterministic approach, we are able to mitigate or to find solutions to mitigate the problem? Could it be a statement that could be made or not? Or as soon as it's stochastic, we cannot manage, and if it's deterministic, we can manage? Yeah, I think.
I mean, models normally you could have in a model both the stochastic and deterministic components.
Yeah. And I don't want to close the door on just throw our hands up if it turns out to be all stochastic, because there's still things you can do. But even if that's the case, you can use these models to more rapidly figure out what wet lab pilot scale experiments you want to do. So you're being very effective. And the reason you want to do that is, let's say we say, okay, stochastic is terrible for ARGs. We just don't want that to happen. Well, then we're going to have to design our systems to make sure that determinism is the prevalent mechanism. And that line of research, you're definitely going to want all the tools you can find to quickly formulate hypotheses, filter them, and work on the most promising ones.
Yep.
Thank you. And maybe to close soon the roundtable, because we all need a break before the award session at 12:30 CET. Francesc, are there some burning questions still in the chat that we can ask to one or two speakers?
Okay. There's a question from Shaya, and it's addressed to Michael Gillings, but I think anyone could talk about it. Thank you for your nice presentation. Any idea on the functioning of these engineered organisms in the presence of locally adapted microbes already present on site in case of field applications?
And to make context, Shaya is also a bioremediation engineer and in a company in the Netherlands, and they look at stimulating bioremediation of field sites.
Interesting question. Usually engineered organisms don't fare well in complex microbial communities. I remember a very long time ago did some work on an engineered rhizosphere organism. Introducing foreign microorganisms to a functioning microbial ecosystem is often quite difficult because there's all of the organisms are already there. If possible, I'd prefer engineering to be in a very controlled environment, like a giant bioreactor or something like that, where you can actually control all of the parameters. And I'd prefer environmental solutions to be driven by evolutionary and ecological principles, where we understand how microbial communities work and we just pull the levers to change them, to speed up or slow down processes. But yeah, I guess it's very context specific as well. I mean, what microbial ecosystem do you want to tweak? I don't really have a solution for you. I'm sorry.
And at the same time, we need, if we would go this direction, we need to maintain the organism in the ecosystem. So all the organisms that you mentioned that are present, they have been selected by the conditions of the system. So to which extent it relates to bioaugmentation as well in wastewater treatment plant, to which extent the inoculum that we feed in the wastewater treatment plant to bioaugment the sludge would remain over time. Maybe locally over some weeks it can work to remediate the problem, to which extent we can maintain the organism in the system. So we spoke yesterday about immigration, extinction, drift, etc. But to which extent we can maintain a specific population inside a well-established ecosystem already? This is the question. So thank you very much, Michael. I don't know if you wanted to follow up.
No. It's it's been a pleasure. This has been very interesting, and as I said before, I could not see my presentation loaded up in the documents, but I have uploaded it. So if people want that presentation, they should be able to get a PDF copy, and I'm more than happy to answer questions offline or via email later on.
Thank you.
And I wish you all the best with your conference. It's really interesting. I've looked at the program. It's a very interesting conference. So.
Thank you so much for your contribution, actually, and all the speakers. Regarding the presentations, I think if you had a pre-recording, the pre-recording should be accessible to everybody. Just a thing for the slides, we would have to ask all the participants or all the speakers to let the slides on. versus the scientific content of the slide. So not everyone wants to have the slide set delivered as is, downloadable to the participants. We
No,
mainly
that's
the videos. Yeah.
That's fair enough, but I did a pre-record as well, which I uploaded. So it's a shorter version, but maybe that could be uploaded and people could look at the details, the references, things like that if they need to. Yeah. Thank you very much. It's been a pleasure.
Thank you so much, and thank you everybody, and have a nice evening, Michael and the others. Bye bye, and we see each other at 12:30. There will be the recreational part with the awards together with Bart and Amit, and the early career awardee and the Arden Lockett awardee, and we will also award the posters before continuing with 2 sessions this afternoon on water and health and the COVID-19, how it impacted wastewater systems. Thank you so much.